Welcome to the Best of Microbe 2017 from the Microbe meeting in New Orleans. I'm Jeffrey Maloy.
And I'm Stanley Maloy.
And today we're going to discuss some of the highlights of the meeting this year.
Before we get going, I want to remind people in the audience, if you have any questions or comments, there's a microphone in the back. We'd love to have you participate in the conversation. And if you're watching us online, please send any questions or comments through the Facebook comment thread. We'll get to them as soon as we can. We have two guests today that Jeff and I thought really represent some of the exciting stuff here at the ASM meeting in New Orleans.
So we have with us Harmit Malik, and Harmit is at the Fred Hutchinson Cancer Research Center in Seattle. And we also have with us Chris Kellogg, who's at the U.S. Geological Survey.
So Harmit not only gave a wonderful talk, but he actually won the Eli Lilly Award, so congratulations. That's a tremendous honor. And he gave a talk that showed why he was a clear winner, which set this aside as one of the highlights of the meeting. And the talk focused on molecular arms race between host and virus. And so part of it focused on that, and the other part was this concept of Paleo Virology. So why don't we start off talking about Paleo Virology? When you say paleo anything, people think you need a fossil record to be able to track it. So how do you do Paleo Virology without a fossil record?
Well, actually, in this case, the idea of paleovirology started with a fossil record, but it's not a geological fossil record, it's more of a biological fossil record. So when the first surveys of the human genome came out, of course, the big debate was about how many genes the human genome encoded, and that was resolved pretty quickly. But what was actually really staggering was the discovery that our genome was chock full of, hundreds of thousands of imprints of these selfish DNA elements or their remnants, which had decayed. And I was particularly struck by the fact that there were nearly 100,000 imprints of what were clearly descendants of previously infectious retroviruses that have invaded our genome and are now basically decaying subject to mutation. So that was in fact the fossil record that first sort of created this concept that we could use that fossilized version of the virus and hundreds of thousands of versions to recreate the history of these viruses.
So, Harmit, are these fossils that you're finding in the genome, are they purely interesting from a historical perspective, or is there some application to studying them?
Yeah, so that's an excellent question. So, of course, the primary interest that I had in studying them was to reveal the evolutionary history of infection that these viruses were, because if you consider what it takes to leave even a single imprint, the virus needs to infect an organism. It not only needs to infect the organism, it needs to infect the germline of the organism. So the cells that produce eggs and sperm, and then it needs to get passed on and fixed in the species. And apparently that has happened not just once, but 100,000 times in the lineage leading to the humans. And keep in mind also that the mutational decay is such that our ability to detect these fossils decays pretty rapidly after about 80 million years. So everything I'm talking about are fossils which we can still recognize. There's probably an equal proportion of dead versions of viruses that we can barely even recognize from a bioinformatic perspective. But most of them are actually decaying, but some of them have not decayed entirely. Some of them encode protein coding genes that we have found great use for. For instance, there's a fossilized version of a virus that still has an intact envelope gene. The envelope gene is what is used for infection. And that version of that protein is actually used for placental function in not just us, but in all mammals. And so that's a case of like the primate genome, having encountered this virus, decided that's an envelope gene I want to preserve and protect from mutation, and has been preserved. And there are literally about thousands of examples of enhancer elements, genes, switches that turn genes on and off, that are all derived from retroviruses. So retroviruses come in built with not just these protein coding genes, but with all of these switches. And as they sit in the genome, they have been basically commandeered to create completely new gene regulatory networks that are a critical part of many pathways in our biology. So I sort of think about them as that this is like a bombardment of the surface of the moon by these meteorites, but occasionally some of these bombardments were very adaptive and were actually beneficial and preserved.
Interesting. So I have to say, the idea that this envelope gene really mediates placental function is nothing short of mind-blowing. It must have been unbelievable discovery. I mean,
It was an entirely, actually this discovery was made by two different labs from the Malik lab and the Heidman lab in France. But what made it even more profound, actually, work that has come out more recently, is that there are 7 lineages of mammals that are now dependent on their own version of syncytin genes. In each case, they rely on a completely different envelope derived from a completely distinct family of retroviruses. So not only has this happened once, this has happened 7 times. And in primates, it looks like we've actually traded up a couple of times, where we've gone after a more recent version of an envelope gene and basically lost a previous version. So it is kind of mind-blowing to think that something as fundamental as how we give birth could actually owe its origins to a retroviral infection.
So you've discussed viral genes. Are there any bacterial genes that are left behind in the human genome?
Actually, when the human genome came out, there were like a slew of papers talking about how many bacterial genes there were in the human genome. It is pretty controversial, but I'm of the viewpoint that there are actually very few bona fide cases of horizontal gene transfer in the human lineage. Now, that's not to say if you cast a wider net across animals, there are many, many examples of bacterial genes. And if you look in ticks and mites, for instance, they have stolen a gene that bacteria used to kill each other to now kill bacteria. So it's a case where ticks, having encountered a protein that bacteria are really good at to kill other bacteria, have now basically become part of their defense arsenal. So this kind of horizontal gene transfer occurs all the time. It just happens that the human lineage has relied more on viruses for its innovation than it has on bacteria.
so one of the things that across microbiology, we use a lot of different techniques. Some people are more molecular biology, some are more biochemistry, and others think genetically. And although some folks don't know the difference between molecular biology and genetics, I think those of us who do genetics know there's a difference, right? And so I tweeted yesterday about your talk, and I said, this is really amazing because most people who look at a lot of sequences, they'll take a molecular biology approach. They'll say, oh, we want to move this part with this part and make these combinations. But a geneticist will think about it a little differently in terms of how they go about it. And I said, you do it like a geneticist. And your answer was, of course, because I am a geneticist. But could you say something about why genetics is so great for doing this versus cut and paste kinds of things?
I think I'm going to offend a lot of molecular biologists, but I'll say it anyway. I think genetics is a really great way to discover something that you had not anticipated. I think molecular biology is a great way, in fact, the only way to dissect something that you've dissected and how the mechanism works. But something like this syncytin gene, you look at it and it's like, this absolutely does not make sense. For that, we needed to have like an expectation of what would you expect for a decrepit gene of that age. And then you realize, okay, this completely violates that. That's a very molecular genetic kind of argument to make. But then what that gene does, of course, requires molecular and cell biology. And of course, the nice thing about this field is that it has people like me who are at the beginning stages of like identifying case studies of these domestications. But then we also have molecular biologists who have basically taken these fossilized versions of viruses and recreated completely active versions as they must have existed 6 million years ago. Of course, we are very careful when we do that because that's how all bad science fiction novels start. So we only do it one gene at a time. But the really nice thing is we can actually discover not just the functions of these dead viruses, but we can also actually discover what were the host genes that they interacted with. And in one case, we actually discovered that a version of a gene that helps protect rhesus macaques against HIV was a version that evolved in humans differently to protect us against this ancient virus. But having done that adaptation, it is now no longer good against HIV. So there are these fitness trade-offs which don't make sense at all, except if you view it in the light of evolution.
So that's fascinating work. So Chris, on the other hand, has done a totally different kind of approach, again, from a genetic perspective, looking at the microbiome of coral reefs. So Chris, what got you interested in this work initially?
Hard to say where to start. So I grew up in the Virgin Islands living on a sailboat because my parents were those people you always hear stories about who decided to run off and then never came back. And so I think living with the ocean as my backyard sort of predisposed me towards marine biology. And I was lucky enough I ended up in graduate school in a laboratory where they were doing sort of molecular microbiology. And that brought together the things I like best, marine biology and genetics. And it all came together. And as I've worked on a variety of things, but have been nice to come back around to coral and doing solidly marine work.
Yeah, you'll always be able to do your work in the ocean where you're most at home, right?
Well, my friends tease me. They said, if you're going to specialize, you really nailed it, because I picked resort microbiology, because if you do tropical coral reefs, it's typically a really nice place.
So we've heard a lot about a lot of depressing news lately about coral reefs and the terrible condition they're in, but you work on a different kind of coral reef than we normally think about, deep sea coral reefs, and you've suggested that those reefs might be a little more pristine. Can you explain why they might be more pristine and what the condition of them is like?
Sure. So the reefs I work on are, I call them deep sea corals, although really it's cold water corals, because where they are is based on a thermal range. So they're happiest between 4 and 12 degrees Celsius, so what it feels like inside your refrigerator. But where they are often are deep enough, so off the continental shelf and hundreds to thousands of meters deep. And so my sort of thought is they're, buffered by being far away and deep from sort of the mess we keep making. So they're sort of, the distance and the depth buffers them from a lot of the insults of, nutrient or sewage pollution or pesticide runoff, or in most cases sedimentation, and even from temperature, because the temperature range in the deep ocean tends to be much more stable than in the shallow surface layers.
Interesting. So, sorry. So how deep are we talking? I know you mentioned it's more about the temperature than the depth, but when you say deep sea corals, are we talking about 30 feet down, or what?
So it depends on where you are. So like I said, because it's just cold, sometimes you can get these coral species in scuba diving depths, which would be 30 meters, and that would be in fjords like in Alaska or Norway or Chile. Most of the places I've worked, they're hundreds to thousands of meters deep, so we need submersibles or remotely operated vehicles to collect the samples.
Wow. So have you been able to go on any of those submersibles to see the reefs?
I have. I've done more than 10 submersible dives in 2 different types of submersibles.
And what's that like?
The first one was probably the trickiest. The Delta submersible literally looks like a little yellow submarine like you would see in the cartoons, and there's room for 2 people. So one person sort of lays in a fetal position looking out a porthole this big, and if that's me, the guy driving it is in the conning tower and he's sitting like this. And it really didn't make me comfortable knowing that if something went wrong, he could pop the lid and maybe make it to the surface, but I wasn't going anywhere because he was between me and the door. Versus the Johnson Sealing submersible has one of those huge acrylic bubbles in the front, and so you and a pilot are in that bubble, and because it's open, you feel like you're not closed in at all. And then there's a separate compartment in the back where there's 2 more people.
Wow. So you don't have an issue with claustrophobia, I take it?
And not at all.
And how long are each of these dives?
Usually the battery power in subs tends to last about 4 hours. So you selectively dehydrate, you do your dive, and then everybody bursts out and runs to the bathroom as soon as you get back to the ship.
But nonetheless, you need to really like your co-inhabitant of the submersible, even if it's just 4 hours.
Yes, you're in close proximity to people, absolutely.
So the deep sea corals maybe provide a great opportunity to get a solid baseline of microbiota that's unaffected by some of the things that are in the populations that are under decline right now, right?
That's what I feel. I don't know if anything anywhere is truly unaffected, but I view them definitely as minimally effective, or as more minimally as we can hope to find these days. And I also like, again, because they're living hundreds to thousands of meters deep, they're in the dark. They don't have the photosynthetic symbionts, the zooxanthellae. And to me, that just takes one more level of complexity out of the symbiosis that is corals. So you can really sort of get a closer look at the coral animal and its other microbes, the bacteria, the archaea, the fungi, the viruses, and try and tease apart what's going on with one piece removed.
Mm-hmm. So if these corals are less affected by surface conditions like climate and tides and sunlight, what kinds of factors are associated with different microbiota conditions in these corals?
So I wouldn't say they're not affected by surface conditions, it's just in a totally different way. So because they don't have photosynthetic algal buddies to feed them, they have to be in areas that have high surface productivity. So certainly any climate or environmental conditions that are going to change that surface productivity, that's their food that comes raining down. And so that's an area that impacts them. And where they live in the ocean, the pH is already different. So they're living under a pH of 7.9 compared to surface corals where we're looking at 8.1.
Is that true of deep sea corals around the globe that they're
in general, most based on the depth range, typically, yes.
Great, okay.
So they are still affected by things, it's just different in how.
Very interesting.
So there's been a lot of discussion about now I'm going to get into an area that may be tricky. There's been a lot of discussion about the USGS budget lately, right? The same way that those of us dependent upon grants from the NIH or NSF are concerned. And so a question is, when you start thinking about these areas, like looking at coral reefs and the correlation with climate change, is this something that you think the USGS is going to really be able to maintain and continue these baselines over the near future?
Yes, because it's so much more than just any one thing. It doesn't just fit into a box with climate. In fact, I kind of laughed when I was asked to speak in a session called Microbes and Climate Change, because I don't feel like I work on climate change. And when we go out to sea, because it is incredibly expensive, to go on an offshore ship and to have an asset, whether it's a submersible or a remotely operated vehicle, that's expensive. It's up to $50,000 a day. So it's not like I just go. We usually have a team of up to 10 to 15 different scientists, and not all from the USGS. Usually we have people also, from NOAA, from several different universities. And so everybody sort of gets together so that we can all go out for 2 to 3 weeks, and you parse out all the different objectives. So yes, we're sampling corals so I can look at their microbiomes, but then I share that coral with someone who's looking at coral population genetics, and someone who's looking at coral taxonomy, and someone who's looking at coral reproduction, all of which have different components of things we're trying to learn. How isolated are these deep reefs? Is there connectivity between different deep reefs? And then there'll be a fish person who's trying to look at them, because the reefs are basically nurseries for certain commercial species of fish. You might have someone looking at the invertebrates. And so we have all these different components that all come together. So we're working to actually categorize the entire ecosystem, of which my part is just one piece. And that ecosystem level is really important, and especially with the deep reefs, one of the main reasons we're out there looking at these ecosystems, mapping them and characterizing them, is in support of Bureau of Ocean Energy Management science. They're not going to lease this piece of the bottom of the ocean for oil and gas drilling if we don't tell them what's there.
So I'm going to make a shameless plug, even though what you do is not all climate change. The American Academy for Microbiology and the American Geological Union just published a little short fact on microbes and climate change. It's really nice, brought in people from a lot of different areas, and you can download it free online. So anybody listening, it's a great read, I think.
It was. I highlighted at the beginning of our session because it covers not just the ocean, but the atmosphere, soils. It covers sort of all of the different areas of the world and how they're connected to climate.
Right. So,
can I ask a question of Chris?
Absolutely.
So what are the types of questions you are interested in with your research, in the sense that are you finding lots of changes in the local microbiome of these coral reefs, and how dynamic are they? If you go in year after year, how much variation do you find from one season to the next?
So that's the trick, because it's so expensive to get out. You don't usually get year after year, and so a lot of times we're literally back at the level of who's there in each coral. And so you start out with the who's there, and then if you're lucky, you start to move into what are they doing. We're not usually at the space, I'm lucky if I can get some biogeography where I get to sample the same coral in a bunch of different places so we can get a sense of regional differences and how that might be linked to food supply or to other environmental parameters. Can't really tell you much about change, but that's the idea of the more I can benchmark these things now, the more I've created that 0 point against which people, me or others, can compare.
Yeah. And in terms of like different coral reefs across the world, is it like like with some cheese cultures, whether you grow them in Vermont or like Washington State, they tend to like have the same starter cultures and they take the same progression pathway? Is it the same with corals, or is it completely a different starter set and you end up with a completely different constitution?
Hard to say. So I think so. It's very clear the coral host determines what its microbiome is. So two different coral species, even if they're right next to each other, have very unique microbiomes. And so I think the coral's determining how it ends up, and I guess you could say they all have the same starter culture because it's the ocean, but then they're very selective of who they keep and how they build their microbiome.
That's clear. Thanks.
So an interesting thing is to say, could you do Paleo Virology with the virus coral connections? which might be a lot more complicated in terms of putting all of the lineages together, but that's the kind of thing that might also lead to some insights that could help respond to problems.
No, I think it'd be interesting because, as I understand it, so the deep sea or cold water corals were the originals, and then you had this, one offshoot that went and shacked up with the algae, like, your weird cousins. And so the more ancient corals would be these deep sea corals. So maybe looking at them is sort of a fossil version of looking at the tropical corals and might get you further back.
Yeah, absolutely. Yeah, and people are now sequencing a number of these coral genomes, and so I think we will have the data set to be able to address exactly these kinds of questions.
Without the Jurassic Park idea, right?
Well, it is even pre-Jurassic in some way, right? If you think about it, but it's actually really interesting how much of what we consider as like our visible biology is actually driven by events that happened billions of years ago at the bottom of the ocean, and even we heard Nick Lane talk about even like our ability to do energy consumption as a proto-eukaryote probably required the unique kind of geochemistry that was like near these hot air vents, etc. So it's really humbling to sort of see how these sort of single events and this unique chemistry opportunities afforded there basically led to all of the biology we see today.
Well, so both of you are things we thought were pretty amazing highlights of this meeting. There's a lot of great stuff, but you alone, either of you alone, was enough to make it worthwhile, I think.
Oh, thank you.
It'd be really interesting from your perspective. What did you think was just a highlight or two of the meeting and why it was so exciting?
Well, this morning I attended a session called "All the Worlds of Phage," and I thought that was just a fascinating session. There were 4 talks on 4 completely different topics. Each of them was fantastic. Since we have limited time, I'll just highlight one of these talks, which was from Paul Turner's lab at Yale, who actually started with very simple evolutionary principles of, bacteriophage target bacteria with very different receptors. And he wondered, through previous work in his lab, he had said, well, if you evolve bacteriophage and the bacteria have to avoid them, he had shown previously that they do so at the expense of making themselves more vulnerable to antibiotics. So he went out into this, he didn't tell me where, but some pond in New England, and discovered this completely new bacteriophage that actually targets the efflux pumps of Pseudomonas. The efflux pumps are what the Pseudomonas does to pump out all the antibiotics that we throw it at it. So your $1 million experiment, the bacteria actually takes 20 minutes to kick out of the cell. And he said that if you basically target these bacteria with these phage, the bacteria now evolve mutations or entirely jettison their efflux pumps, making them extremely vulnerable to antibiotics. So it's this really profoundly simple evolutionary idea. And then he demonstrated that if you do a combined therapy with just an antibiotic, the bacteria are totally fine. Their biofilm totally resists this. If you do the phage thing, the bacteria are also fine, but you combine them, you trap the bacteria where it can't actually win simultaneously against both, and the biofilm disappears. And in a spectacular example of this, they actually got FDA approval to treat a human patient who had a pustule of Pseudomonas near his heart, which he had basically been told like there's nothing we can do from a medical perspective. They got approval to treat this patient with this cocktail of phage and antibiotic. They had tried the same antibiotic before and it hadn't worked, but in combination with the phage, they had like complete resolution.
Wow.
So this is a case where the paper is actually not out yet. Peer review can take some time, but this was already covered by Carl Zimmer in New York Times and places like that. So this is a profound example of, we've heard all along about the promise of phage therapy,
yes,
but this idea that phage therapy takes bacteria to a place which is really bad for their ability to resist antibiotics really renews my hope in that combined therapy would be really fantastic. To me, that session was like the highlight for me so far.
That sounds really cool.
I should say that's very interesting because in parts of Eastern Europe where people have used phage therapy a lot, very often they use combination phage and antibiotic therapy, but they just didn't understand why.
Yeah. In fact, in a different talk, they did metagenome sequencing for one of these commercially available Russian phage cocktails, which had previously, I actually don't know how these were made initially, and they've been testing these out in Bangladesh, and they've been able to molecularly dissect exactly which phage was more important against the E. coli that caused diarrhea. So we're really at the point, because of all of this power of sequencing approaches, where we can go into habitats, including our own gut, where we would have no ability to discern actually individual organisms. We can discern those organisms, we can grow them up in culture, and we can treat people. It's not actually very different from, I work at a cancer center about immunotherapy. This is pretty much like dark ages of cancer therapy, which is even 10 years ago. We had no idea what we were dealing with, but now people can take cells out, grow up the exact immune repertoire you want, and put them back into people and get result. We're at that stage, I think, or just on that precipice of that stage, which phage therapy, which is very exciting.
Yeah. Just one other thing. We are knee-deep in beautiful studies on phage that helped us understand the biology of phage. And what's happening now with phage therapy relies so much on that. So when sometimes people talk about applied research versus basic research, to me, this is why you need that continuum.
Yeah, it's a false dichotomy.
Absolutely.
I mean, this session was a perfect example of that, because it started off in space that you would be very tempted to say is like evolutionary microbiology and laboratory experiments to say what's happening. And it slowly progresses, but each step is like a huge transition, and within 3 steps you're basically treating human patients. So I actually think that all basic sciences is applied by definition.
Nice.
So Chris, what about you? What got you excited?
I really liked at this meeting, it seemed to be a sort of shift in programming where several of the sessions, and so the one I was in, microbes and climate change, but also one I saw on Friday, which was sort of about wildlife conservation, it's like they were taking these sort of broad crosscuts where you had people talking from a bunch of different, what I would consider biomes. So someone talking about, an ocean system, a freshwater system, a soil system, maybe atmosphere, across one common theme. And I just found that really incredible, because you pick up so much more than when you make it very narrow and it's like everybody's talking about something in the ocean, or everybody's talking about just in this host system. Spreading it out and really showing sort of the breadth that you can encompass within some of these topics, that's all still microbiology, has been really fascinating to follow.
That's great. So what about you, Jeff?
So before I talk about something that I thought was a highlight, I want to remind the audience here and the audience watching online that we would love to hear from you and hear some of your most interesting talks, your highlights of the meeting, and any questions you might have for our guests as well. So one of the talks that I found really fascinating at the meeting was yesterday, Chris Woods talked to us about using host markers for infection in order to improve our rapid diagnostic techniques. So his group found that there are under 100 genes, there's a very small subset of genes that you can use to predict very accurately whether or not a person's infected, whether they're infected with a virus or a bacterium. And the reason that this is really important is we've talked a lot about this meeting as well. There have been a lot of sessions on antibiotic resistance. Mm-hmm. And so one of the problems with antibiotic resistance is when a patient comes into a hospital and says, I'm not feeling well, I have an upper respiratory infection. A lot of times it's a viral infection, but doctors are often afraid that if they don't prescribe an antibiotic, the infection, instead of resolving, will get worse, and they'll end up with a worst case scenario in the hospital. So we overprescribe a lot of antibiotics, and it's really important for us to be able to figure out rapidly whether an infection is a bacterial infection or a viral infection. So the work of Chris's group really is important it allows us to see whether someone's infected with a bacterium or a virus much earlier than we start to see symptoms. So we can see right away if it's important to prescribe antibiotics for that person. So I thought that was a really interesting talk, and there's some really cool applications as well. So another thing that he talked about was his group is starting to look at proteomic signatures of infection. And so if we could find 30 proteins or even less that are host proteins that are indicative of bacterial infection, then you could potentially come up with a paper-based assay. So you could have a tissue that when you sneeze into, it turns blue if you have a bacterial infection, which would be a really cool way of rapidly telling whether someone's infected and keep that.
And you could use it in the field as well. You didn't even need to do it in the hospital. Yeah.
And if it was really good, it turned blue if it was gram positive and red if it was gram negative.
Well, so I'm wondering how hard it would be to translate some of those things then to coral diseases, because the problem with coral diseases is corals aren't good at telling you symptoms. They have, sort of two signs. They turn a different color or they drop dead. And that doesn't give you a lot of range in terms of figuring out what the problem is or determining whether it's bacterial or viral or something else. And so if some of this could be converted into a field-based thing that applied to a system like that, that would be incredibly helpful for basically, park or reef managers to try and manage some of the coral disease outbreaks, because the faster we could figure out something was coming before things just start dropping dead in front of you, the more time we have to try and manage that.
We have a question now from the audience.
A question more so, it's an observation. As far as what was my favorite here at this session, I've been coming to ASM conferences for a long time. I think my favorite is that these talks are now in the exhibit hall, and as well as all the other track hubs that have the rapid fire and poster talks. So you're not running around all the different halls, and you actually get to get a taste of everything. That's been my favorite thing.
That's great.
I think that's a great point. So this year there's so much going on, it's often hard to we've talked some about how there's conflicts between, you want to go to two talks at the same time, and so this year there's been a lot of increases in ways to view talks from nearby locations on TV screens.
Although I was not able to resolve that conflict because the one talk I really wanted to attend was going on the same time I was giving my talk, and I thought it would be inappropriate if I left my own talk to go attend this other talk. So I was relying on people tweeting about the other talk, which a lot of people were really good at, and that felt like I could actually keep up with the other talk. But yes, it was somewhat of a comedic situation.
So kudos to our live tweeters.
Yes.
It's a good problem to have, too many good talks happening.
Absolutely.
So, for me, if you go to a 20,000 foot level, something I really like is science goes through phases where everybody's talking about what they've done, they're trying to move into the next period. But it's almost like we're in this revolutionary phase of science where we're thinking about what's next. How can you have that approach to use this in coral reefs? How can you take the Paleo Virology and use it to apply for these other things? And so it's exciting because you can learn a lot of new things that may be outside of your field and you wouldn't see. But you're hearing about where things are going, what people are thinking, not just what they've already done. So
I agree. I think there's I echo what Chris was saying, that the fact that we are talking about multiple systems even in one session, you as an outsider, you actually leave that session really inspired by the principles, even though each individual story you might not remember the intricate details, but you really sort of can appreciate what are the principles that are driving this field. And that's actually one of the things that the program committee has done very successfully this year, to not get completely in one versus the other. I'm not, without criticizing, I have attended ASM for many years now, and of course technology drives a lot of the biology. And when sequencing technology was in its heyday, we were playing bingo to hear the words 454 sequencing or Illumina sequencing, and you could actually plot it. But what was not actually clear was people were doing a lot of that, but the principles weren't coming because people were just so excited by what they could do rather than thinking about okay, what does that mean? We are now at the point where we are thinking, oh, what does that mean? And that's really exciting.
So that reminds me, we're talking about a lot of paradigm shifts in microbial sciences we've heard about this year, and one of the things that I think was a big paradigm shift for me in the way that I think about some of these classic sequences that we've looked at and thought about for forever. You talked about in your talk, you discussed the idea that for a long time we've looked at conservation of sequences as an indicator of importance, right? And your work now looking at sequences and kind of reevaluating them from a hardcore genetic perspective is giving us new insight into how that might not be the case.
Yes, from an evolutionary perspective, what I said is completely not a surprise to evolutionary geneticists. But I think as molecular biologists, so much of our focus has been on the conserved parts of proteins or genomes, because if it's conserved in evolution, it must be important. And that's absolutely true. But what has been sort of considered true, which is false, is that the most rapidly evolving parts of the genome mustn't be important, because if they were important, they would have been conserved. In fact, these represent the ways that these genomes or proteins are actually adapting to the challenges that they face. And so what I simply presented was some case studies where my lab has actually focused on the most unconserved parts of the protein, and those actually turn out to be the most important in terms of specificity changes. So I think these are just like, the nice thing about like an umbrella program like the ASM is you get all of these ideas coming from different disciplines, and hear these talks, and like I already have 3 ideas just in the last 2 days about things I should be doing in my lab, my own dogmas and like how I should try to get rid of them. And so that's the nice thing is like you can come in and everybody can feel slightly humbled by what they don't know.
So one of the other talks that virtually everybody seems to have loved is Kate Rubin, right? Talking about microbes in space.
Speaking of taking a 20,000-foot view.
They're a little higher than that, I think. but it dealt with things like concerns about leaky spacesuits and biocontaminants in space and things like that aren't the kind of things we normally think about. So it was a lot of fun. And if anybody out in the audience wants to know more about that talk, and if you missed it, she was also recorded live on TWIV. So you can watch her TWIV interview, which is really very, very nice as well. I might say this guy right here was on TWIV 4 years ago?
Yes, many years ago.
Oh, several years ago.
You have a better memory than I do.
Yeah, and that's also a very good interview. So anyway, that's nice because that leaves a little paleo record of the way you've thought across time, right? So.
Yes, and that's not on video, so you can't actually see how much I've grayed since then.
Well, so a lot of good stuff. One of the things that we haven't covered in our talking about the best is everything that's after this point in time through the end of the meeting. And there's a lot of other things that I think are really exciting. One talk I'm really interested in is how low levels of antibiotics, that is concentrations below inhibitory concentrations, how they might select for resistance, which is an important problem because right now we're kind of swimming in a sea of antibiotics wherever we go in the world. So that's an example of kind of a new thinking that'll be really fun and integrates a lot of different aspects from basic through practical. But a lot more to go, and we want to thank you for joining us today. It was a really fun conversation. And
so thank you for joining us at the Best of Microbe in 2017. Again, this is Harmit Malik, and this is Chris Kellogg, and I'm Jeffrey Maloy.
And I'm Stanley Maloy. We also want to thank all of you who are out there in the audience, and all of you who are listening online. You actually have a lot of friends out there that are joining you in terms of the numbers. So ASM is a great place. There's a lot of exciting stuff. We hope you come back, join us again for Microbe 2018 in Boston. In Atlanta. Don't go to Boston next year. Go to Atlanta. Thanks very much, folks.
Thank you for having us.
They tell you to put your chin right there.
I became an ASM member in 1981.
95?
Since 2008.
I joined ASM in 1974.
Been an ASM member for 41 years.
I first became a member in 1989.
Seems like all my career.
I'm willing to bet it's a decade.
My first ASM was 1993. I still have a picture of me holding the program open, sitting on the hotel room bed, really excited because for the first time ever I saw my name in print associated with microbiology.
When I was a graduate student at Purdue in 1980, it was the thing. I mean, if you were going to be a microbiologist, you joined the ASM. It was just really that simple.
ASM is very special to me because I became a member in 2002, and when I first attended the meeting at Salt Lake City, Utah, I met my PhD thesis mentor, Dr. Arturo Casadevall. And so ASM kind of introduced me to the PhD world.
Definitely the most important thing is that ASM has provided me with a graduate fellowship. So they've helped support me during my graduate training In addition, I've gotten a lot of networking opportunities, and I've met a lot of really great people through ASM.
And we can share our work and connect, and they can teach me things that I don't know, and I can teach them things that they don't know. And just that partnership and working together, that's what scientists do, to share your work. And it's the most exciting thing to learn about new projects.
ASM has actually done a whole lot of work and given me the kind of exposure I wouldn't have had if I'm not a member of ASM.
Many brains are better than one.
Great thing. It's really member-driven organization. I love that. I've always loved that.
Could make the bigger the better, the more the merrier.
You want to do microbiology, become a member.